Hi class, and welcome to your next video on membrane transport. Now we're going to talk a lot about how things are going to cross the membrane and exactly what does that mean for our cells. So let's go ahead and get started. So how do essential nutrients enter cells and how does waste enter and leave cells are going to be two of our essential questions that we're going to really want to consider and think about as we go through this presentation. So first off, we have our whole membrane here. So within our membrane, we have a lot of components to our membrane. So I would like you to either take a screenshot and put this in your notes or to draw it. So let's talk about the pieces to the membrane though. So on the outside here, we have the glycoproteins, we have some carbohydrates, and we have some things such as the extracellular fluid and matrix. Now, the glycoproteins and the carbohydrates are going to help identify the cell. They're going to be things that really help different molecules identify this is a certain type of cell. It's a blood cell, or it's a bone cell, or it's a nerve cell, or it's a plant cell, or it's a fungus cell. It's really going to tell different molecules what is this type of cell. Then we have the extracellular matrix is going to provide support and the communication with other cells. We also have collagen, which is going to be that support, okay? So within that extracellular matrix, that support comes from the collagen, which is a form of protein. And our surface carbohydrates are going to be what is going to communicate. So that's our identifiers. Remember, I just told us about our identifiers. Those are going to be our main identifiers. Inside of our cell, then, we're going to have internal support and transport. So our cholesterol and our filaments of our cytoskeleton and some of our proteins are going to help provide some support, and they're going to help get things through. So those proteins are going to help get different types of molecules through, and the cholesterol is going to help provide some structure and some framework. So the cell membrane is also the gatekeeper. It's a selectively permeable, meaning it only allows certain things to pass through, but other things need help. So the phospholipid bilayer is 2 layers, and it only certain things can get through the phospholipid bilayer. So some molecules can pass through the membrane unaided, and some molecules need the protein channel to cross. So things that are very large are going to need the protein channel to cross. Certain molecules that cannot diffuse through that polar phospholipid bilayer are going to go through the protein channels. So let's talk about the selective permeability and exactly what is that. So the phospholipid bilayer, like I said, only allows certain molecules to pass directly through based on their chemical properties. Small molecules pass through easily, and ions and large polar molecules need a protein channel. So if it is a polar molecule, it will need to go through a protein channel. All of those large molecules need those protein channels. They need a little bit of help. Okay, so our membrane, though, is the gatekeeper. It's going to be the guy that says you can come in or you cannot come in. And it's based on the charges, the size, all of that. And it's going to be based on what the cell needs. Diffusion. So all of this is passive transport, meaning it requires no energy. And you can actually do this on your own very easily. Molecules always travel from an area of high to low concentration across a membrane. So it's also called moving down the concentration gradient. So we're moving from high to low like we see in the picture here. And small molecules can diffuse in and out of the cell through the membrane. And so what's going on here is you're actually, if you see here, we have a high concentration right here. Like I said, we're going to move from high to low. It's going to evenly kind of spread out. If you were to do a short little experiment, what would happen is if you took a glass of water and you put a drop of some kind of dye into it, let's just say food coloring, and you let it sit in there, at first it's going to be very, very, very concentrated in one area, and you're going to see where that drop goes in. And then over time, it's going to diffuse out, so it's very evenly spread out. And maybe over an hour, 2 hours, 3 hours, those molecules are going to move around in that cup, and eventually it is all going to be spread out very, very evenly. This is diffusion. It requires no input energy. We do not need anything. Now, facilitated diffusion, we still need no energy, but we're using protein channels to help those polar molecules and large molecules cross. So the only difference in facilitated diffusion is we have to use the protein channels to allow those larger molecules to cross that barrier. So diffusion and facilitated diffusion use no energy, but in facilitated diffusion we must use those protein channels. Now, how the cell membrane maintains homeostasis. Homeostasis is the cell's happy set point with good concentration of both water and solutes. And we always want it to be the same concentration inside the cell and outside the cell. They are always moving solids and water across the membrane. Everything is always moving. It is never staying stagnant. So if the external environment becomes too full of solids or becomes too full of water, the cell must maintain homeostasis. So what's going to happen is it's going to move that water one way or another. It's going to move towards the area of the higher concentration of the solid. So if it has too much water in one area in the cell, it's going to move it towards the outside of the cell where it has a higher concentration of the solid. And if it has a higher concentration of solid inside the cell, it's going to move it towards the higher concentration inside the cell. So when we move this water, we call it osmosis. Osmosis is just the diffusion of water across the membrane in response to solute concentration. And water follows solute. An easy way to remember this is salt sucks. Salt is going to suck the water one way or another. So if we have a higher concentration of salt inside of a cell, the water is going to move towards the salt concentration. So salt sucks that water. Now, let's look at this. So I want you to copy down these terms, the hypertonic, isotonic, and hypotonic, and I'm going to explain what all of these mean. So in a general cell where the concentrations are equal, you're going to have an isotonic cell. So if we have 1% of solute inside the cell and 1% of solute outside the cell, we have an isotonic cell. The water is constantly moving in and out at an equal pace, but that cell is happy. It's maintaining homeostasis. Now, if we have a higher concentration outside of the cell and a lower concentration of salt inside of the cell, the water is going to tend to move towards that higher concentration, which is outside the cell. So it's going to leave the cell, and that is in a hypertonic cell. And so the water leaves that cell, and in an animal cell, that goes through crenation. When the cell shrivels up in an animal cell, that is called crenation. In a plant cell, when the cell wall comes away, or when the plasma membrane comes away from the cell wall. That is called plasmolysis. So the opposite of that is when we have a higher concentration inside the cell and a lower concentration outside of the cell. So the water is going to come into the cell. With a plant cell, this is actually where the plant is happy. The plant loves this. It wants this. So the plant is going to gather all this water into its central vacuole, and the plant is going to be turgid. This makes your celery crispy. This makes your lettuce crispy. This is what's good for a plant cell. For an animal cell, this is not good though. The animal cell will then burst because it does not have the cell wall to help with the structure. So it'll burst because of this. This is due to osmosis. The next form is active transport. Active transport is requiring energy in the form of ATP to move against the concentration gradient from low to high. Remember, we talked about moving from high to low. Now we're going to move from low to high. And the membrane proteins are necessary to actively move the molecules against their concentration gradient. So usually we're going to disperse out from that high concentration. Well, now we're moving them against what they usually want, and we're going to go towards that high concentration. And we want to have a high concentration. And this is going to require ATP because this is different than what should naturally happen. Okay, so I want you to really have an understanding of all the cells. Make sure you got great pictures, especially of the plasmolyzed, turgid, and isotonic crenation in the cells, and plant and animal cells. Make sure you know the difference between active transport and passive transport. And we will talk about this in class tomorrow.